Good evening, everyone. My name is Caroline Jean Fernald. I'm the Executive Director of Harvard Museums of Science and Culture, where our mission is to foster curiosity and a spirit of discovery in visitors of all ages, enhancing public understanding of and appreciation for the natural world, science, and human cultures. With this mission in mind, I am delighted to welcome our in-person as well as our Zoom audience to tonight's lecture co-sponsored by the Department of Organismic and Evolutionary Biology, the Harvard Museum of Natural History, and the Harvard Museums of Science and Culture. We are honored to have Professor Molly Przeworski from Columbia University with us tonight, who will discuss the promises and pitfalls of genomic trait prediction. To learn more about upcoming events, I invite you to visit our website, hmsc.harvard.edu. We have several educational and fun program opportunities for adults, teens, and children. I will now turn it over to Scott Edwards, Professor of Organismic and Evolutionary Biology, Curator of Ornithology, and Alexander Agassi, Professor of Zoology in the Museum of Comparative Zoology, and Chair of the Department of Organismic and Evolutionary Biology, who will introduce our speaker. Thank you.
It's great to be here, and it's my great pleasure to welcome you to the John M. Prather Lecture Series, which is an annual celebration of an exceptional scientist in the field of biological sciences. The series is inspired by a bequest from John McClellan Prather. Prather was a zoologist and high school biology teacher who earned his bachelor's and master's degree from Harvard University in the late 1890s. And his PhD from the University of Chicago. The departments of organismic and evolutionary biology and molecular and cell biology come together each year to host this series as a recognition of an extraordinary biologist and educator. We're thrilled this year to have Professor Molly Przeworski here today to present the public lecture in her Prather Lecture Series. Molly is the Alan H. Kempner Professor of Biological Sciences and Systems Biology at Columbia University. She earned her BA in Mathematics from Princeton University, her PhD in Evolutionary Biology from the University of Chicago, and was a postdoctoral researcher at the University of Oxford. She was a researcher at the Max Planck Institute for Evolutionary Anthropology, and then a faculty at Brown University, and then the University of Chicago before joining the faculty at Columbia University in 2014. Molly has earned countless awards and recognition for her groundbreaking research to better understand how the fundamental processes of evolution, natural selection, recombination, mutation, operate in humans and other animals. As an early career scientist, she was awarded the Sloan Research Fellowship, an early career award from the Howard Hughes Medical Institute, and the Rosalind Franklin Young Investigator Award. Her accomplishments have more recently been recognized with being elected as a member of the American Academy of Arts and Sciences and the National Academy of Sciences. These honors reflect her truly innovative research that focuses on the population dynamics of natural selection. What I most enjoy about Molly's work is her clear exposition of complex and sometimes controversial topics. Her work explaining even to professional audiences the nuances of genetic testing and what it can and cannot tell us about our identities and our fates is authoritative and requires us to look under the hood at the inner workings of genetic algorithms and methods, some of which are first presented as foolproof. Now let's give a warm welcome to Molly, who will speak to us about reading genetic tea leaves. Thank you very much.
Thank you very much, Scott, for this introduction, and to the Prather Committee for this incredible honor, to all of you for joining me in this venue tonight. In case you're wondering what this is, I was playing with Crayon, which is the free version of the deep learning software that is meant to generate images because I can't draw, and I wanted to evoke the image of reading genetic tea leaves. And I had this 1 hour long attempt to get it to understand what I meant and decided I should have drawn it. But here it is for you. So today I thought I would tell you about a topic that I'm imagining you've seen increasingly in the news or in ads, which are DNA tests that help to predict all kinds of things, in particular about your life. So here are some examples where companies might propose to sequence your genome or part of your genome to inform you about your cancer risk, or maybe your susceptibility to cardiovascular disease, or what allergies you might suffer from. Or here is an example in which gene sleep might inform you about whether you're actually a morning person or what skin regimen to use. You might also have seen headlines about using your genetic code to predict something about how well you might perform at school. There are yet other companies that propose to make these kinds of predictions not for you, but for your newborn or even your embryo prenatally. And then on a maybe lighter note, companies that propose to scan your genome for evidence of romantic chemistry, or to help you choose the ideal wine for your palate. So my hope is basically to try to address the question of what this might possibly all mean. What are these companies purporting to do when they promise to read your future in genetic tea leaves? Which of these predictions are accurate and why? So to try to demystify a little bit of how genomic trait prediction works, whether it is for your risk of cancer or for your choice of wine. And by and large, what I'm going to argue is that there are areas, notably for medical prognosis, where this kind of genomic trait prediction may turn out to be useful. But by and large, genomic trait prediction is both much less informative than these ads and headlines would suggest, but also a lot less mysterious. In addition, though, to outlining some of their pitfalls, I do want to highlight some possible promises. And there, what I'm going to argue is that, in my opinion, actually, their most useful application may be not to predicting our futures, but instead to predicting the future of species that are really important in ecological responses to climate change, in particular to help prioritize conservation efforts. So, to start, I think it's useful to divest ourselves of a notion that actually runs throughout biology and has been extremely helpful, but can also be misleading in some contexts. And that is the notion of a wild type, the notion that there's a typical way in which a species presents itself and operates in nature. So here, for example, is a wild type Drosophila melanogaster, the fruit fly, the workhorse of many labs in molecular biology. And here is a mutant in which a mutation has altered the genetic code in a particular location of the genome, leading to these legs instead of eyes. So this concept of a wild type is meant to encapsulate how development is meant to occur in this mutant development gone badly awry. And of course, this kind of mutation is highly deleterious. This poor fly is not going to fare well, is not going to leave descendants. And so the mutation is going to be rapidly lost from the population. And even if it were to recur, will never be anything but rare. So this way of conceptualizing things in terms of wild types and mutants lends itself, I think, quite naturally to a scientific approach in which you introduce such mutations, or maybe you screen populations for rare occurrences of such mutations, and then you use them as major perturbations of the system from which you can try to then reverse engineer how the system is built, in this case, to learn about how limbs are specified in development in the fly and in mammals. So importantly, this approach works because these mutations are of large effect. They're often sufficient to drive the whole process, as in this example. And these are the kinds of traits that we all learn about in high school biology, so-called Mendelian traits that are caused by single mutations and tend to lead to a discrete change in the trait. But in point of fact, as Darwin and many, many others since him have emphasized, what you see for most traits, if you look at natural populations, is not a wild type and some very rare mutants, but graded variation and often quasi-continuous variation. And that's true in many things that you might think to look at, shell color polymorphisms within a single snail species, or color patterns in frogs, or colors of morning glories, or even the extent of coral bleaching among coral colonies, a trait that I'll return to. It's also true in humans. So some traits like polydactyly or terrible diseases like Tay-Sachs or cystic fibrosis are almost always due to single mutations. But most traits that we might think to consider, height, total cholesterol, behaviors like startle reaction or risk aversion, susceptibility to diseases like type 2 diabetes or various forms of cancer, actually vary continuously in the population. And they're not caused by one mutation. They're caused by many, many mutations and environmental effects. So consider the example of height. Here's a simulated distribution for women in the US based on actual data from 10 years ago now. And you can see there's a distribution of height in the population. It is not distributed as a wild type height and rare mutants. In fact, it looks much like the US soccer team. Now, of course, there are other sports like basketball that might benefit from very tall players, but basketball players are not mutants either. They're simply draws from the tail of this distribution here, women who happen to be very tall. So there are gradations of trait values in humans and other species rather than a wild type and rare mutants. And these differences in height among people, say among the people in this room, are due to all kinds of environmental effects that we encountered in utero and growing up. For example, whether we had access to enough food. Importantly, though, however, differences in height aren't entirely due to environmental differences among us. In fact, in wealthy contemporary societies, most of the variation in height is due to genetic differences. Now, when I first encountered this idea, it seemed counterintuitive to me because I was also aware of the fact that height shows huge secular trends, which are clearly the effect of the environment. So here, for example, is a map of world heights comparing the average height of women at the end of the 19th century to what it was a century later at the end of the 20th century. Okay, and you can see that women increased in height on average all over the world, but in some places much more than others. So in particular, if you focus your attention, I don't think I have a pointer actually. If you focus your attention in South Korea, then women gained an astonishing 20 centimeters on average over 1 century, so about 8 inches on average. If you look at North Korea right above, they actually gained on average about 10 centimeters, okay? And that is clearly not for genetic reasons. It's because of the different political and environmental conditions in which women found themselves in North and South Korea since the 50s. Okay. But importantly, differences in height being mostly due to genetics is not a statement about variability at any given point in time. Is a statement about variability at any given point in time. It's not a statement about the mean over time. So it may be that the distribution of heights in South Korea looked like this at the end of the 19th century, shown in purple, and maybe like this at the end of the 20th century, shown in gray. There was a 20 centimeter increase in the mean height, but the variability remained much the same. And importantly, women such as myself who find themselves on the shorter end of that distribution, or women who might find themselves taller than average, find themselves there largely for genetic reasons. So at this point, I think it's important to emphasize that the fact that variation in a trait is largely due to genetics does not mean that environmental interventions don't make a difference, right? The obvious example is myopia, the risk of which is largely genetic, and yet, of course, I can wear glasses or get LASIK and not be myopic anymore. Deprive people of basic nutrition, they will be less tall. But the fact that variation in height is in part due to genetic differences does mean that somewhere in our genome, there must be information about how tall or how short we end up on average. What about for other traits? Well, here's an estimate of the proportion of the trait variance that can be assigned to genetic variance. And height is the tall bar here, because as I mentioned, variation in height is mostly due to genetic differences, and that's not true for most traits. For most traits, most of the variability is not genetic. Nonetheless, you can see that for pretty much any trait, including anthropomorphic traits like height, medically important traits like vitamin D levels, monocyte counts, but also behaviors like age of first birth, that first column there, there is a genetic component. And all that means is that somehow, by mechanisms that are almost always unknown, some of the variance in the trait is due to genetic differences among people. In that sense, the fact that variation in a trait has a genetic component is to a large extent an uninteresting statement. Even the odds of getting hit by a bus, which might seem like the quintessential accident, the risks of that are partly genetic because getting hit by a bus is partly due to our level of absent-mindedness, and some of us are more absent-minded than others, and that has a genetic component. But these kinds of results do establish that not only are most traits continuously distributed within the population, some of the variation that we see arises from differences that reside somewhere in our genomes. So what do these genetic differences look like? Well, for the past couple of decades, we can get at that quite directly. We can read out the sequence of A, C, T's, and G's that they're comprised of, and we can count up where they differ. And these sequencing efforts have confirmed that not only does almost any phenotype you can think to look at vary within the population, there's also a tremendous amount of genetic variation within humans, like within any other species. So consider, for example, the 2 copies of, say, chromosome 1 that you inherited, one from your mother and one from your father. Okay, if you align these 2 chromosomes side by side, you would find that most positions are identical between the maternal and the paternal chromosome, but a subset will differ. And in humans, that subset is on average about 1 in 1,000 base pairs. 1 in 1,000 of these letters differs between any 2 chromosomes. Okay. Now, given how long the genome is, that translates to about 3 million single base pair differences between any 2 genomes. Okay. The exact rate at which they differ depends a little bit on the recent genetic ancestry of the individual. So humans with, for example, recent African ancestry tend to harbor more genetic diversity than people with recent European ancestry, and so they might differ in a position in every 800 base pairs rather than 1 in 1,000. But overall, in the human population, there is a difference every 1,000 base pairs or so. That's between a pair of chromosomes, but different pairs might differ at distinct positions. So here, for example, is a larger study of human populations called the 1,000 Genomes Project, and this is where they sampled individuals in the globe. And you can see that they've sampled all over the world, and they found a great deal of genetic diversity, specifically 85 million different base pair variants. And of these, 8 million were pretty common. So that ended up meaning that they found a common variant, a variant that was common in the population, and there was more than one letter, so more than one version in the sample, one in every 400 positions. Okay. So what does this variation look like in practice? Here's a representation of a tiny region of the human genome, just to give you a sense of it. Each column here is a site that differs in the sample, and each row is a person color-coded according to their genotype, so what mutations they carry. If they carry 2 copies of the common letter or allele, then that's in blue, 2 copies of the rare allele, that's in yellow, 1 copy of each is in red, and if it couldn't be determined, it's in gray. Okay. And you see that even in this tiny stretch of the genome of maybe thousands of letters, and in this sample of 47 individuals, there's already a tremendous amount of genetic variation. So who are the wild types here and who are the mutants, right? At the phenotypic level, it doesn't really make sense. And you can see that at the genetic level too, it doesn't really make sense. There are actually no wild types and mutants. There are only mutants. So now, as it happens, this sample is comprised of 2 population samples, 24 people who self-identified as African American, and the next 23 people were sampled in Utah who had recent European ancestry. And as you can see by eye, there are some differences in the frequencies of the different genotypes as you move along the sequence, but they're subtle, and most of the variation is shared. And this is, again, typical of what you see in the genome. If you consider different populations sampled around the world, then most common genetic variation is found within any one population, and only an additional 15% is found between populations. So another way to view this is illustrated here, 4 common variants, okay? So the overlap in these circles is reflective of the degree of overlap in common genetic diversity. And you can see that there's a circle that overlaps a little bit less, and you probably can't read that, but these are individuals that were sampled, Yoruba individuals that were sampled in the capital of Nigeria, okay? And as I mentioned, they harbor, on average, more genetic diversity. But overall, you can see that even though these samples were gathered in Bangladesh, in England and Scotland, in Los Angeles for individuals of recent Mexican ancestry and so forth, the dominant pattern is definitely one of shared common variation. And the reason is, loosely, that most humans around today are actually quite closely related on an evolutionary timescale. And despite the fact that they've occupied the whole globe, we're continuously mixing throughout this time period. So there's been very little opportunity for genetic differentiation. So where does that leave us? We have a huge amount of genetic diversity in humans within any given human population, and that genetic diversity contributes to variation in numerous traits. In other words, if I were to sequence, say, all the individuals in this room and measure our heights, then I'd get a wide distribution of heights. It would be roughly bell-shaped, and I'd find tons of genetic variation, and I'd know that somewhere in our genomes must be some variants that affect height. But how do I go about associating those two? How do I go about associating variation in height to specific locations in the genome? Well, these days in human genetics, this is usually done by an approach called a genome-wide association study, or GWAS. And GWAS aims to identify the loci in the genome, the positions in the genome that contribute to variation in complex traits like height. Now, the idea is actually pretty simple. You pick a location in the genome, indicated with this sliver, and then you regress or relate variation in the trait to genotypes you see at that position. So you put the height of individuals carrying AA there, and the individuals carrying AG at that position there, and the height of people carrying GG at that position there. And then you just fit a line, okay? And if there were no relationship between that genetic variant and height, the line would be flat. If the line increases or decreases, that's indicative of the fact that that genetic variation is affecting height. Okay. Moreover, the slope of that line, the steepness of that line, is indicative of the effect of that allele. Meaning, if I were to go in and substitute AA for AG, how much would I increase height on average? Okay? And the scatter around that line is due to the fact that I've only considered one position in the genome, and lots of other positions matter, as we'll see. And there's also environmental effects on height that I haven't considered. So that's what leads to the noise, to the scatter around that line. So that's basically the idea of a genome-wide association study. But now, of course, you have to rinse and repeat at all the different positions along the genome. Okay. So here, for example, is the result of a recent such study for height, which amazingly considered the height of over 5 million people. Okay, and what I'm showing you here is the number of variants associated with height in any given stretch of 100,000 letters or base pairs. Okay, so you can ignore the details here. What I want to draw your attention to is that there are a huge number of variants that have been associated with height. Almost every stretch of 100,000 letters has one, and often several, on average 2. Okay, the authors estimated that there are 12,000 independent signals here, 12,000 positions in the genome in which changing a letter would change your height on average. Okay, so the genetic basis of height involves many, many variants, and each of these variants has a tiny effect on average, changing height on average by 1/10 of a centimeter, so 0.04 inches. Okay. So importantly, these are not the mutants that people study in model organisms. These are very, very mild perturbations to the trait. Okay. So a person's height reflects a sum of environmental effects and then the effects of these many, many, many genetic variants. And that suggests a natural way of estimating someone's height based on their genetics, which is just to sum up the effects of all the alleles that someone carries that influence height. Okay, so suppose we did that. Let's consider our 2 copies of chromosome 1 again, now represented by these 2 rows of circles. And let's label every position that affects height in white here. And let's label the alleles in terms of whether they increase your height or decrease your height. So shorter or taller alleles, okay? And now we can just go through and count up. Let's pretend this is my genome, although I'm aware that I probably do not carry that many tall alleles. And the numbers in the bottom here are the estimates of this allele substitution based on the GWAS that we have conducted hypothetically, okay? So I go through and I say at this first position, I carry a T, so that's plus 2 millimeters. At that second position, I carry a T, that's plus 0.5 millimeter. Next position, I carry 2 Ts, plus 6 millimeters, and so forth. When I'm done doing this with all 12,000 positions in the genome, I have a genomic prediction of height. And that really is, unfortunately, all there is to genomic trait prediction, okay? So at this point, it's important to note 2 things. First of all, even though variability in height is mostly due to genetic differences, there's still a big effect of the environment on differences among us, say whether I ate enough in my early childhood. And we don't have environmental effects in this predictor, so we can only do as well as genetics allows, right? If half the variability in height is due to genetics, we can predict at most half the variability in height. So if we go back to our example of North and South Korea, then we are not going to predict a 10 centimeter difference between women in North and South Korea because that is not in their genomes. The second point I want to make is that prediction is not what it seems, right? So I think a lot of us have this implicit model of what genomic trait prediction should mean, and that is that we understand something about how that genetic variant affects the protein, affects the cellular phenotype, affects development, and therefore affects height, say. In practice, for almost all complex traits, we are very, very far from that. All we really have is this massive correlational study. Okay, so all this to say, sort of dispel from your mind the scene in Star Trek where they scan your DNA and they say, oh, this mutation is doing this to your cells and that's why you've turned purple. We don't have that kind of understanding, right? In practice, for complex traits like this, we almost never know why that variant is associated with that outcome. Now, that doesn't mean that we don't do well at prediction, because actually you don't have to understand how things work to do well at prediction, right? For example, consider Netflix's suggestion of what you should watch next, which is accurate. In my mind, it's actually spookily accurate. And yet, of course, it doesn't understand, hopefully, anything about you, right? So this is genomic trait prediction. And the specific exercise of adding up all the causal alleles weighted by their estimated effect sizes is this polygenic score that some of you may have heard about. Now, I'm presenting these applications as if they were somehow developed for humans or in human genetics. Actually, most of these statistical approaches were developed not in human genetics, but in agriculture, where a highly related approach has been used with great success for decades to predict traits like milk yield in a cow or drought resistance in rice. Okay? So in humans, what might polygenic scores be used for? Well, one context is to detect people at risk for disease before they show any symptoms, to screen them more regularly or maybe give them prophylactics. So here, for instance, is a figure from a very influential paper looking at this polygenic score, this genomic trait prediction for cardiovascular disease. And on the y-axis is the prevalence of cardiovascular disease in different sets of people. And on the x-axis is sort of the score that they got in terms of their genomic trait prediction, where people predicted to be at greater risk are towards 100 here. And you can see that people predicted to be at greater risk do turn out to have a higher prevalence of cardiovascular disease, and therefore, for example, one might imagine wanting to give them statins. This has also been reported for other diseases, notably cancers, such as breast cancer. As you may know, some women are at high risk of breast cancer because they carry single rare alleles of large effect, and as a result, they often get screened more regularly. Other women may be at risk, similarly high levels of risk, because of lots of variants of small effect, and they too may similarly benefit from more regular screening. So this isn't my area of expertise, so I'll just add that to my understanding, there's still an ongoing debate about how useful these scores are really going to turn out to be beyond things we already can assay, like family history or running various biomedical tests. But in principle, at least, there's clear utility in being able to predict people's risk of disease before they actually get the disease. Our prediction ability, however, is not restricted to biomedical traits. In principle, we can go through this exact same exercise for any trait where some of the variability is genetic, given a large enough study. So for example, people have conducted numerous genome-wide association studies for years of education, a trait for which it's estimated that maybe a third or 40% of the variability is associated with genetics. And they've conducted these studies because years of education is something that you often fill out when visiting the doctor, so it's available for very large numbers of people. Most recently, it was conducted in 3 million. They did a GWAS similar to the GWAS for height in 3 million people, and they built a genomic predictor of years of education, and then they went and they predicted actual years of education in different cohorts. So here is an example where they've predicted where they've built a genomic predictor, which they call polygenic index, but it's actually a polygenic score. It's just been renamed. And what they're showing here is how well this genomic predictor predicts actual years of education in the AdHealth cohort. And the AdHealth cohort is 20,000 people that are of European ancestry for reasons that I won't go into, but in other regards are nationally representative. Right. And so if the prediction were perfectly accurate here, all the points would lie on the line. So the prediction is far from perfectly accurate. Nonetheless, they can explain about 16% of the variability in years of schooling. That's what this R-squared value is telling you. So what about the other 84%? Well, some of it is that we don't know all the locations in the genome that predict educational attainment. And a lot of it is that there are obviously environmental effects on years of education. So you can't predict individual years of education all that well, as you can see from how noisy this plot is. But you can predict group behaviors a bit better, how people with a low or high polygenic score do on average. So one thing you might do, as in this paper, is you might treat the polygenic score, the genomic predictor, as informative about what they termed, and I bolded I quoted them, but then I bolded it myself, DNA-based indicators of the propensity to succeed in education, okay? So they use this genomic trait predictor, and then they went and examined for people who have a high score in blue and people who have a lower score in tan, what math classes they took through high school. And that's what they're plotting here. They've ranked math classes by their level of difficulty, and they're showing you that the sort of overall gestalt is that people with higher scores take harder math classes, okay? And the point of this paper was actually to say that in schools that are socioeconomically disadvantaged, this is more so true, whereas in schools that are more socioeconomically advantaged, even students with lower polygenic scores, with lower predicted years of education, end up taking more advanced math. As I'm going to discuss, however, I think this interpretation is misleading. And the reason for that is that contrary to the way that DNA-based indicator of propensity to succeed in education might sound, these predictors are not, in fact, based mostly on your genetics. Now, to understand that, we need to dig a bit deeper into what the estimated effect of an allele on a trait really reflects. Naively, you might think, well, a variant carried by a person influences, say, some aspect of brain development, leads them to absorb information more rapidly, and therefore to be better at school. Okay, we call those direct genetic effects of an allele carried by an individual on that person's traits. Okay, now it's a bit of a misnomer because direct genetic effects need not be direct. So here's an example. There's a variant in a gene called CHRNA3. That gene is a nicotinic receptor, okay? And it influences the risk of lung cancer. But actually, in this case, we know, and it's rare that we know this much about mechanism, in this case, we know that it has this effect entirely mediated through its effect on how many packs of cigarettes you smoke a day if you smoke, okay? So this is an example of a direct genetic effect that is in fact entirely mediated through a behavior. Nonetheless, direct genetic effects are causal in the sense that if I were to modify the variant in CHRNA3, I would influence how much the person smokes if they smoke on average, and therefore I would, in fact, influence the outcome, risk of lung cancer. Okay, unfortunately, associations detected in a genome-wide association study reflect a lot more than these kinds of direct genetic effects. They reflect indirect genetic effects, ways in which the genetics of family members and peers have shaped the environment of the person. They reflect assortative mating patterns of the parents, and they reflect population structure confounding. And I don't have time to delve into all of these, so today I want to highlight in particular this problem of confounding, okay? What does it mean? Suppose you did a study of reading ability in an elementary school, and you found shoe size to be associated with reading ability, okay? That is not, you will be surprised to hear, because shoe size affects reading ability. It's changing a kid's shoe size is not going to alter their reading ability. It's because of confounding, because, say, older kids have bigger shoe sizes on average and also tend to have greater reading ability. There's an underlying cause of both reading ability and shoe size that makes them associated. Now imagine you do a genome-wide association study of bagpipe playing ability. I learned from Wikipedia, preparing this talk, that people play bagpipe all over the world, not only in Europe, but in North Africa, West Asia, and so forth. But it's obviously a common custom, in particular in the Scottish Highlands. And for the purposes of this example, I'm going to stipulate that bagpipe playing is more common in Scotland than in England, even though I'm happy to be corrected on this point. So say you do a genome-wide association study in a set of individuals, some of whom have recent English ancestry and others of whom have recent Scottish ancestry. Genetic differentiation between these two samples is going to be very low. But there are going to be some subtle differences in allele frequencies, right? With millions of common variants, there are going to be positions in the genome where variants are more common in people of recent Scottish ancestry than English ancestry simply by chance. So now you do your genome-wide association study of bagpipe ability. You find maybe some genetic variants associated with musical ability. But you're also going to find any variants that happen to be a different frequency in Scotland and in England and that are therefore associated with Scottish cultural practices. For example, you might find genetic variants for red hair. Not because red hair is causal for bagpipe playing, but because genetic variants that underlie natural red hair color are more common in settings in which people are likely also to have learned to play the bagpipe. So that's the problem that goes by population structure confounding. When people's genomes are associated with particular environments, it becomes really hard to completely disentangle the effects of their genomes from those of their environments. We have statistical tools to do so, and we use them, but they're not perfect. So let's return to the traits years of education. The Genome-wide association study has sampled people in different zip codes. In this world, people with different ancestries are not randomly distributed across zip codes. So when you sample different zip codes, you get different ancestries. And because different zip codes provide access to more or less high-performing schools, include parents who are more or less wealthy, and so forth, what zip code you're raised in also affects your years of schooling. And that generates an association between genetic variance and educational attainment that is not causal, but confounded by differences in environments that are associated with differences in ancestries. So for years of education, this has actually been considered explicitly. And for example, what the authors of this study found is that even restricting their attention to people of recent European ancestry in the United Kingdom, the effects that were being estimated in the Genome-wide association study likely reflected, in part, geographic variation in socioeconomic status across the United Kingdom. And this is, again, my bolding. Another study, again focusing on British of European ancestry, found an effect such as that, an effective population structure confounding, not only on years of education, but also on BMI and fat mass, hypertension, and lung function. So all this suggests that when we do a genome-wide association study and we build a polygenic score, we aren't just basing ourselves on the blue effects here, but also all kinds of other effects that are associated with genetic effects, but are not effects of the allele carried by a person on that person's trait, and may in fact not be genetic at all. So how can we go about figuring that out? How can we estimate what fraction of what we're detecting as a genetic effect is in fact genetic? So one way to tease these things apart is to make comparisons only within a family, say by looking at how the kids departs from their parents, okay? So loosely, what that approach allows you to do is to control for the environment and almost anything associated with the genetics of the parent. So I'm not going to go into detail here, other than to tell you that comparisons of these kinds of genome-wide association studies done within families to the usual genome-wide association studies allows you to understand how much of the predictive power is actually coming from direct genetic effects versus all the rest, okay? Now, one thing to remember is that precisely because when you do this within a family, you only capture direct genetic effects, if you build your genomic prediction based on these family studies, you may not do as well as predicting the outcome, okay? Predicting bagpipe playing ability based on genetics is pretty hard. Predicting it based on recent genetic ancestry is less hard, okay? Predicting years of education based on genetic effects is hard. Predicting it based on your zip code is unfortunately not so hard, okay? So confounding muddies interpretation, but it can help predictive power. So a few years ago, we set out to test how much of the predictive ability of genomic trait prediction was due to direct genetic effects versus these other types of effects, okay? And by we, I mean Hahamanesh Mostafavi and Arbel Harpak, two former members of my lab who are now faculty. So what I'm showing you here for a number of traits is the prediction accuracy, the ability to predict based on a standard genome-wide association study versus on one of these studies conducted within families that we matched in various ways. And I want you to focus on the squares here because that's what we see on average. And each row here is a different trait that they considered. So if the prediction accuracy were due only to direct genetic effects, then the two approaches would give the same result and they would lie on this line at 1 here. Okay? When the standard approach does better than the approach within families, that's a sign that something other than direct genetic effects is contributing to our prediction. So if you look at natural hair color, you can see that the prediction accuracy of the two is the same. This is on 1, okay? And so what's that saying is that the effects that we picked up in the genome-wide association study are direct effects. That kind of makes sense. We don't expect things like zip code to affect your natural hair color. They may well affect your unnatural hair color. If you focus on height, you see that a standard genome-wide association study does slightly better than a family genome-wide association study. And other papers have suggested a small contribution of a factor other than direct effects. I'm not going to go into it, but I just want to mention that it has to do with the fact that parents tend to be of similar heights. Now here's years of schooling. So as you can see in this case, the standard genome-wide association study does a lot better. In fact, on the basis of this study and a number of others, we know that only about a third of the predictive power comes from direct effects, and the other 2/3 come from some mix of other effects. So maybe the decomposition looks something like this. We don't know. We know that the blue is about a third, and there's at least some green. Okay. More generally, what becomes clear is that for many traits, particularly traits like income or age at first sex or fluid intelligence, a polygenic score, a genomic trait prediction based on your genome, is capturing a lot that is not, in fact, in your genome. So why does that matter? For one, I think it lends a new interpretation to these kinds of studies, which are proliferating, which is that if this genomic trait prediction for, quote, the propensity to succeed in education is actually mostly picking up your zip code and aspects of the environment that children find themselves in, we might interpret the fact that kids with higher scores take harder math classes differently. Okay. More generally, my hope is that all of this has helped you to decipher what such predictions mean, and more importantly, what they don't mean. So, all this might seem a bit disappointing, and I'd forgive you for wondering why I thought to tell you about this evening, and let alone why I might work on them. And I actually asked myself the same question several years ago now. I originally set out to learn about genomic trait prediction because I was interested in using these approaches to learn about trait evolution in humans. But then I became kind of disillusioned about their limitations. And so I started wondering about context in which confounding is fine, in which the thing that matters is to make a reliable prediction, and we might not be so bothered that whether it's based on entirely genetic information or whether it's absorbing some other aspects that tend to be associated with genomic information. And it occurred to me that one area would be if we could make predictions about ecologically important traits in the response to climate change. So for the last 5 minutes of my talk, I want to tell you about our first steps in that direction using coral bleaching as an example. So I need to make a series of disclaimers at this point. First, I'm not a climate scientist, and I probably know much less than most of you about climate change. Second, I'm not a coral biologist. In fact, the closest I get to the species I work on is by staring at the ones in my lab aquarium, shown here. But thankfully, I have amazing collaborators and foolhardy lab members who have been patiently educating me. So I want to particularly highlight the contribution of Zach Fuller, a former postdoc in my lab, without whom this project would not have taken off. So as you likely know, reef-building corals are the foundation of incredibly diverse marine ecosystems. They're home to about 4,000 species of fish, hundreds of other species. They also sustain economies across the globe through tourism and fishing, and protect the shorelines against storms and floods. But the ocean warming caused by climate change is leading to the breakdown of coral reefs and basically threatening these ecosystems worldwide. In fact, modeling projections of coral reefs call into question whether they will persist till the end of the century. So a key reason for that, for the breakdown of coral reefs, is that they're critically dependent on algae, on dinoflagellates, that they have a symbiotic relationship with algae, with these endosymbiotic dinoflagellates, from which they derive most of their nutrition. Okay. And the high temperatures are disrupting this symbiotic relationship, causing the colonies to lose their symbionts and bleach, lose their pigment. These bleaching events increase susceptibility to disease, stunt growth, and often lead to colony death. Unfortunately, the mechanisms underlying variation in bleaching susceptibility remain poorly understood. And as a consequence, conservation and restoration efforts cannot yet effectively prioritize their efforts. We just basically don't yet know where to direct our efforts. And so this suggested to us the possible utility of a genomic trait predictor for bleaching susceptibility. And that's what motivated us to try to work with this system. So we decided to explore if the tools I've been describing to you for humans could also be applied to corals. Basically, if we could build a predictive model for bleaching susceptibility. And for that, we focused on a stony coral, Acropora millipore, shown here in a picture taken in my lab tank, because a previous paper had shown that within this species, at least some of the differences in susceptibility to heat were due to differences among coral genomes. Now, corals are not model organisms. They do not benefit from the resources of human genetics. So we had to build all the genomic resources to do this. But the hard part is actually the sampling. So our collaborators at the Australian Institute for Marine Sciences spent many, many days, many months, in fact, diving to sample the collection that I'll tell you about, originally 240 some samples from 12 reefs along 300 kilometers of the Great Barrier Reef of Australia. And then examine different aspects of bleaching for each colony sample, including a so-called visual score, like what color they appear, basically, as well as other measures. And they also collected detailed environmental information about 40 variables, things like salinity, depth, and so forth. So meanwhile, we collected genome-wide sequences, the genome sequences, for 240 samples. And when we do that, what's nice is we get the genome of the corals, and then we also know what symbiont types they carry. And so confirming what people in the coral community had reported before, we found that corals that carry type D were less likely to bleach, were more likely to be found healthy than those that carried type C, as shown here. We can also run a genome-wide association study. And we basically do the same thing as what I described in humans, except for now we're not doing it for height, we're doing it for visual score or other aspects of bleaching. And from this genome-wide association study, we can build a genomic predictor of bleaching. And now we put it all together, the environmental information and geographic information, the symbiont type, and the coral polygenic score. And we predict the variation in bleaching in samples that we have not included to train our model. And so this is what I'm showing you here. And this is basically akin to the polygenic scores for cardiovascular disease I showed you earlier, where corals in the highest decile here are those that we predict to be less bleached, okay? And even with this small, very small, in fact, sample of 240 corals, we already have a predictive ability in terms of being able to discern which corals seem to be at less risk. And here I would argue it's okay that we're using something that is almost surely confounded. It's okay that we're combining genetic and environmental information. Of course, ultimately, we want to know what factors are most predictive and why. We want to actually intervene in the bleaching, and we're working on that. But in the short term, I would argue that it's already useful to be able to identify which corals are at greater risk, simply to be able to better target some conservation efforts. So with this in mind, our collaborators at Ames have now collected 3,300 coral samples, about 800 from each of 4 species that co-occur along the Great Barrier Reef over, in fact, 1,500 kilometers. We're in the process of sequencing their genomes and basically repeating our efforts to improve our predictive ability and identify which subset of corals might be at most risk. And we're also interested in mapping those genetic effects, or especially interested in mapping those genetic effects that might be shared among species and therefore might be nice targets for intervention beyond this species complex. So to conclude, I hope to have demystified a little bit of what genomic trait prediction is. In particular, I wanted to highlight that not all traits are created equal. Predicting your risk of breast cancer based on direct genetic effects is one thing. Predicting your schooling based on your genetics and your zip code is another. It's obviously up to you whether you want to send your DNA away for a company to tell you which wines are best suited to your palate. But maybe the next time that Scientific American suggests that someone can predict your political affiliation from your genome, you will be less impressed. So also, I wanted to highlight that as it becomes more and more straightforward to collect genomic information, I think that actually its greatest promise may be in applications outside humans in our attempt to predict the responses of species to climate change and hopefully to direct conservation efforts accordingly. And finally, I wanted to thank again the people whose work I have the honor of presenting tonight, including 2 people who are currently in the lab and working on the coral data. Thank you.